Hello, I'm Florian Boecker. I'm a postdoc at the crop bioinformatics group at the University of Bonn in Germany, and I want to talk about AHRD, our program for protein function transfer and evaluation. We want AHRD to be applicable to whole proteomes. For that reason, it has to be fast and scalable. We want to also annotate not only geoterms but also descriptions, and we do that by looking at sequence similarity search results. But of course, these can have problems. You can't always just take the top result. You always have to look at the whole list of results, and also have to take into account that you can have proteins in there where the annotation is actually not really desirable. So here in the description you see uncharacterized protein. You don't want to annotate that. So AHRD has a solution for that. It filters out these descriptions. It also ignores words that are very common to descriptions in general, and scores the descriptions based on alignment quality, the frequency of the interesting words in the descriptions, and because AHRD can use multiple databases, we also distinguish between databases where we have a high confidence and a low confidence. We do basically the same for geoterms, but with the geoterms we also look at the evidence codes of the annotations to have another avenue of deciding whether an annotation might have been transferred to a protein in the past and it's not the original. So the top scoring result is then used to annotate our example protein here and to see how well our prediction actually did in comparison to the ground truth. We see here that the description managed to match 3 out of 4 terms, but for the gene ontology annotation we only matched 1. But of course, with the gene ontology, you always have to keep the graph of the gene ontology in mind. And as you can see here, if you look at the small part of the molecular function tree, with the zinc ion binding prediction, we also implicitly annotated metal ion binding. And with the DNA binding prediction, we also implicitly matched nucleic acid binding. Our prediction was just a little bit over specific. So to get an evaluation metric for protein function prediction that keeps this all here in mind, we came up with our semantic similarity Go score. It uses an information content of each geoterm to derive an most informative common ancestor of a pair of geoterms, and this can then be used to calculate precision in a recall by taking the information content of all prediction terms and dividing them by the information content of all most informative common ancestors with the ground truth. And for the recall, we take the information content of the ground truth and divide it with the information content from the most informative common ancestor with respect to the prediction. So we validate our evaluation metric by performing an artificial dilution series of our ground truth set, which I talk about later. This method was shown first by Plusinen et al. He's from the Finnish group that is behind the prediction tool Panzer. And the rank correlation between our Go score and the signal level of the ADS is 0.969, and the FPS score for false positive sets is 0.164, which both are in line with some of the top performing metrics in their paper. And protein function evaluation is of course always dependent on what to use as ground truth, and for that we take SwissProt and get rid of as much proteins as we can where we think that they don't have the annotation given to them originally, but they have been transferred from other proteins. And we also try to get rid of as much redundancy in the set as possible by looking at the descriptions again and also at the sequences. We now can show you our results. We annotated the 1,000 proteins with using AHRD and some competitors, and we ordered these predictions by their score. And as you can see here, only AHRD and Best plus SwissProt are able to annotate a significant number of proteins with a perfectly matching annotation. in terms of coverage, only NetGo is able to best AHRD, but NetGo does so by trading a lot of the quality in the predictions. We, of course, also annotate descriptions, and here AHRD has the highest number of perfect predictions and also the highest coverage. So AHRD was always fairly easy to use, but we now made it even easier by integrating it into a Snakemaker workflow. Snakemaker is a workflow management utility that uses Conda to manage the dependencies, the software dependencies we need. The user only has to input peptides and FASTA files, and the workflow downloads the necessary information from databases and also does the searches, gets and builds AHRD and runs it, and then spits out the annotations. This is able to scale from a single bacterial proteome on a laptop to dozens or hundreds of eukaryotic proteomes on a cluster. And this is really useful because for sequence similarity, the best method to actually increase the quality of your annotations is to get more original information in, so new database releases. And in order to benefit from new database releases, you must be able to re-annotate your proteomes fairly often. And with our new workflow, that has become very easy and very fast. So we also have a talk about the same topic on this conference, and you can find AHRD on GitHub, and of course also our new workflow. Thank you and goodbye.